Hello, this is Murray Gans from the Scanning Electron Microscope lab here at Eastfield College. Last year for Christmas, my wife gave me a t-shirt that says the following: there are 4 stages in life. The first stage of life, you believe in Santa Claus. Second stage of life, you don't believe in Santa Claus. Third stage of life, you are Santa Claus. And the fourth stage of life, you look like Santa Claus. Apparently, I am in the fourth stage of life. So, here we go. Oh, Christmas tree, how lovely are thy branches. That's a good question. This is a branch from my Christmas tree at home. This is actually a Fraser fir, I'm told. This is just a good old point and shoot picture. Here we're using the dissecting scope, and I really like this picture of this. The greens are just gorgeous. And what you can see is the leaves coming off the stem, and you can see where some of the needles have actually fallen off the stem, which is kind of cool. Here's a closer look at the needles that are on the stem, and you can even see some little some sap from the pine tree here on the base of the leaf. And this is where a leaf was attached, or a needle. Needles are leaves. When I first saw this branch, I thought, well, there's little pine cones on the tip of the branch. But then when I looked more carefully, what I realized is that those are where next year's branches are going to sprout out. So this is a terminal bud, and it's got a place for 3 different branches to come out. So if you look right here, you can see we've got the 1, 2, 3 that are broken open. So that's why you see these on the very tips, and that's kind of cool. Again, you can see the needles off the tree, and you can see where they were attached. Here's the top surface of the leaf, and you can see these little pits. The blue color is kind of an artifact of the dissecting scope, where the light is reflected out, but it's still kind of a nice looking picture. This is a cross section of one of the stems, and you can actually see the pith, which is the center here, and then you can actually see the growth rings on this little stem, and obviously the sap on the outside here. We'll see more of that here in just a second. Here's another picture of a growth ring with some of these rays that connect the growth rings. And here's another picture of the growth rings. And again, this thing, as these branches were drying out, they're actually oozing this pine sap. Here's a picture, an up close picture of the pine sap. And what I did was then I took a small needle and pushed it into the pine sap and pulled back. And when I did, it just came out like this. I thought that was actually kind of a cool thing to see. So now on to the electron microscope. Here's a close-up. These are the 3 little terminal branches there. You can see some pine needles here. You can see where pine needles were attached and came loose. Here's a picture of that, the outer surface of the pine needle here. You can see those little pits that reflected blue so clearly before. Here's a close-up of the stem. You can see the underside of the pine needle here, and then again, places where pine needles have broken off, or where I actually pulled them off. And here's a close-up of where a needle would attach. And right in the center of that is actually, you can see a little bundle of vascular tissue. So here's a close-up look at a couple of little drops of sap. This is a stem over here. This is actually the pine needle, and you can see those little drops of sap attached there. And just like we told you in previous webcasts here, the bright things are just dirt. The dirt seems to glow when you put it in an electron microscope. So here's a cross section of a stem. What I did is took the stem and I cut it longwise, and you can see the center pith area here, and then you can see more of the conductive tissue down in this area. And something kind of weird, we'll see this again in other pictures. These little like blocks of something, and I have no idea what they are, but they keep showing up again and again. They seem to be solid. We'll show you a picture in a second. So in this picture, you can see these little blocks of something. Again, I don't know what they are, but you can also see 2 other things, or a couple of things. You can see the pith down here, which is this center part, and then you can see cells up here, which are part of the xylem, which actually are called tracheids. And these tracheids have kind of something weird. They have these little holes, and these are called bordered pits. And a book I got at the library told me that when water is being conducted back and forth, if an air bubble should get trapped in one of these cells, the bordered pit has a mechanism that actually seals that cell off so it doesn't break the water flow. You can also see some different types of tracheids here. You can see their structure, which kind of holds them open, which is really pretty. Here's a closer look at some of those tracheids again. Just really looks like the trachea, like your windpipe. Here's a cross section. This is at 210 times magnification. Where again, this is in the cross section of the of the stem here. Not sorry, a transverse section of the stem here, and you can see all the cells lined up, most of them going in this direction, with a few in the rays going here, and these little bordered pits everywhere, and that water would go through those pits. Here's a closer look at some of those bordered pits. Kind of cool. This is at 552 times magnification. Even closer look at 1,200 times magnification. Even closer look at 1,000 times magnification. And guess what? Even more at 1,540 magnification. I just was kind of amazed at these. By the way, down here on the micrograph, you can see the distance from here to here is 30 microns. So the distance from there to there is only 3 microns. So these little holes are actually less than 3/1 millionths of a meter in size. When I was looking at a close-up view of the little beads of sap on the leaves, I did notice that there was something else on this pine tree, my little Christmas tree at home, and that there's a fungus growing there, and you can actually see some of the spores from the fungi I've indicated with the black arrows here. And a closer look at some of the spores, and then this is actually a little hypha. This is what makes up the body of the fungus here. So my little Christmas tree, I've not only bought the Christmas tree, I also bought a fungus for Christmas. Here's a cross section of the stem. And you can see I've cut right through again the pith here. You can see the xylem which conducts the water there, and then here you can see I've cut through the base of several leaves, and so you can see how the leaves attach. Here's a closer view of that, and then the last one here, and here the leaf goes off this way, and you can see some of the conductive tissue here. So from the Scanning Electron Microscope Lab at Eastfield College, happy holidays.